Hi, everyone. Welcome to our workshop. We're really glad you're here today. A very warm welcome. My name is Katie. I'm the Best Practice Manager at EMQN. If you're just meeting EMQN at ESHG, EMQN is an EQA provider, but we're also a community interest company. And so we reinvest into our community, and one of the ways we do this is by developing best practice, which is why you're here today. So the plan for today is that we're going to have 2 talks summarizing the recommendations for the hereditary breast and ovarian cancer and inherited cardiac disorder testing guidelines. After each session, we'll have a Q&A where I'll have a microphone. So if you do have a question, raise your hand, and I'll either come over to you, or there's a standing microphone to the left of you where you can ask a question, and the authors of the guidelines will answer them for you. And then after this, after both presentations, we'll have a mix and mingle kind of thing where if you haven't had an opportunity to grab some food or drinks, you can do that, and you can also continue any conversations with the authors that are here today. So I have a few housekeeping items to go over before we get started. Just a general disclaimer that this talk is being recorded. So if you do ask a question, it will be on a recording that we're going to disseminate to our membership, and it will be on our YouTube and LinkedIn as well. And I think that's it today. So I'll pass over to my colleague, Nikki, who will be introducing our speakers.
Thanks, Katie. Let's find the button. No. Hang on. There we go. It's all right, I'll do it that way. So my name's Nikki Wolstenholme. I'm the Science Director at EMQN, and I used to be a clinical scientist myself up until 2015, so I know where everyone's coming from in terms of both quality and also the diagnostic difficulties we all have. So today I'm going to just introduce our speakers. So the first speaker I'm going to introduce, Stacey, the first thing I'm going to do is give a shout out to Stacey. Stacey's not actually talking today, but she's one of the 3 main authors of the HBOC guidelines. Stacey's also done a poster, so I just wanted to give a quick shout out to Stacey that she's done a poster, so if you want to have a little review of the guidelines, there's also a poster down in the poster exhibition room. So the 2 people who are going to speak to you today about the hereditary breast and ovarian guidelines are Trudi McDevitt and Miranda Durkie. So Trudi has a BSc in Biological Sciences, an MSc in Clinical Pharmacology, and a PhD in Molecular Mechanisms of the cancer, I can never say that, cachexia. She's a clinical scientist in molecular genetics since 2000. She's got a diploma from the Royal College of Pathologists in 2002, and she's the principal clinical scientist and co-lead of Molecular Genetics Division in the Department of Clinical Genetics at the Children's Hospital in Dublin. She's a member of the Cancer Variant Interpretation Group in the UK, CANVIG, Steering and Advisory Group. She's the first co-author of the EMQN Best Practice Guidelines for genetic testing of hereditary breast and ovarian cancer, and it has been quite an ordeal to put this guideline together. We had an awful lot of information, and Trudi has done a sterling job of helping organise the writers and the subgroup to actually bring all this massive information down into a guideline that we hope the laboratories will find very useful. So if we move on to Miranda. Miranda's also a BSc in molecular biology and an MPhil from the University of Sheffield. She's got her HCPC, Clinical Scientist registration. She's a Fellow of the Royal College of Pathologists since 2019, and she's the Deputy Head of the Rare Disease Genomics Service for North Eastern Yorkshire, and covering 5 specialist services and core rare disease work. But on top of all of this, Miranda's a very busy person. She's also co-lead of the Cancer Variant Interpretation Group in the UK, CANVIC, Steering and Advisory Group. She's the ACGS representative on the Cancer Genetics Group in the UK, and she's a member of 2 ClinGen variant curation expert panels, so very busy person, and also author of the UK ACGS variant classification best practice guidelines. So I'm going to pass over, first of all, to Trudi and Miranda. So thank you both. It's an amazing task.
Thanks very much, Nikki. Okay, so I'd like to start by, it's great to be able to present these guidelines, so thanks very much to EMQN for the opportunity. And I'd like to start really by, okay, is that better? Yeah. So I'd like to start by acknowledging the huge team effort that went into these guidelines. Actually, I forgot to bring my glasses. So there's a massive team effort. By the first co-authors are in green. They're Norbert, George, Samantha, Kathleen, Peter, and Rachel. Big contribution from that group. Katie and Nicola, thanks so much for all your support throughout this effort, and especially to Katie, who was there right from the start, through all the ups and downs, through COVID, etc., to get it over the line and published in March this year. We had great help from Helen Hansen and Claire Turnbull in the UK for their clinical input and advice to these guidelines. And also the document wouldn't be where it is in terms of being refined if it wasn't for all the feedback that we received. Dr. Michael Morris of SynLab International, members of the EMQN community, over 400, and manuscript reviewers for the review and feedback. It was a challenge to get these documents done because you can see there was 17 years since the last ones, and there's been obviously an explosion of information and knowledge, and everything is really mushroomed in those 17 years. So we ended up with a massive document, as Nicola said, and due to the constraints of the publication, we ended up doing several rounds of cutting it back. So it was actually probably, close to 10 times the size that it is now. So huge team effort. So we're going to go through them. I'm going to cover some background and give a high-level tour of the guidelines, and Miranda is going to take us through a bit more in-depth look at some of the recommendations. So background, so just a quick fly through some of the main changes since 2007. So obviously big changes in the referral pathways, genetic testing, variant interpretation and reporting. And for example, for referral pathways in 2007, most samples received in labs came via clinical genetics. They were mostly ascertained based on personal and family history, based on various risk assessments, such as the Manchester scoring, whereas now in 2024, laboratories can receive samples from lots of different pathways. Some of them are listed here. Genetic testing then in 2007 was mostly BRCA1 and BRCA2 by using a pre-screen such as DHPLC and Sanger, and the protein truncation test was still widely used. CNVs by MLPA, but now of course it's NGS gene panels, either large panels or just selecting high-risk genes. And we have somatic tumor testing, CNVs by MLPA, and increasingly by NGS. Variant interpretation, of course, loads of work in this area, as you can see by all the guidelines since 2007, both rare disease and cancer-specific ones. Reporting in 2007 mostly concentrated on diagnostic and predictive test results, whether a pathogenic variant was present or absent, and in the case of males, really just concentrated on implications for relatives. Now we have some scenario-specific wording to help laboratories, and Miranda will talk about those in a little bit more information in a little bit later. So high-level tour of the guidelines. So the purpose of the guidelines is to encourage standardization of HBOC testing throughout diagnostic laboratories by providing guidance on testing, interpretation, reporting that covers advances in knowledge, technology and practice, and is aligned with established genomic testing practices that are now a feature of diagnostic laboratories. They're international guidelines, so they acknowledge policies across different jurisdictions as well. They're aimed at scientists involved in diagnostic HBOC testing, so those providing a minimum service of, for example, BRCA1, BRCA2 and PALB2, to those providing a comprehensive multi-gene NGS service, and also to laboratories, many of them pathology laboratories, who are offering somatic tumour testing and HRD testing. For the structure of the guidelines, then, we have a big supplementary material section, which was necessary because of the constraints for publication in the European Journal of Human Genetics. So a lot of important information went into the supplementary material. There are 7 sections, with the recommendations mostly spread between 3, 4, 5, and 6, and the largest number of recommendations, as you'd expect, in the reporting section. Supplementary materials, just going into this in a little bit detail, just take you through these. So we have 2 tables that we spent quite a bit of work pulling together from 23 large studies. We pulled collated information on gene-associated cancer susceptibility risk. So, for example, you can see an excerpt here for BRCA1. The table covers all the genes you can see listed here that are associated with a definitive associated cancer risk. So we provide information collated out of the studies on the locus, the OMIM gene number, the main select transcript, mode of inheritance, and the pathogenic case prevalence details from the studies, the relative cancer risk, and the absolute cancer risk. Grey shading indicates if there's a high associated cancer risk, and if there's no shading, it's the moderate risk. So this is for Table 1, and we hoped that these tables will be useful for labs for informing multi-gene panels, informing decisions on putting together multi-gene panels and selecting relevant genes. Table S2 then are a list of those genes that you often find involved on panels for HBOC testing, but for which there's conflicting evidence or a less defined risk. I should say that for BRCA1, BRCA2 and PALB2, as well as the ovarian and breast cancers, we also looked at other associated cancers. We only looked at it for those 3 genes, just for the purposes of the guidelines, because those are the 3 genes where most of the pathogenic variants are identified. So this Table 2 follows the same format as Table 1 in providing risk information. We provided Table 3 as a summary of all the studies that we looked at. So there are 7 shown here, and there were 23 that we pulled the information out of, mostly large case control, prospective, retrospective, and literature reviews. We also provided a quick reference guide in the supplementary materials. This is Table 4. There are 50 recommendations, and in this table, the grey shading indicates a must variant, so it's essential, and where there's no shading, it's a should variant, so highly advised. The recommendations are organised in terms of the section that they're in, and for some of them, there is a linked reporting recommendation. For example, you can see this one, recommendation 5 under genetic testing also has an associated recommendation in the reporting section. So where that happens, we've indicated it there. So I'll move on to the recommendations and hand over to Miranda.
Okay. Thank you so much, Trudi. Don't panic, I am not going to go through all 50 recommendations. And so thank you both to my co-authors and EMQN for helping facilitate it. But also, before I start, I just feel like we need a little pat on the back for getting to that nice round number of 50 recommendations. It wasn't planned, but it was just very pleasing. We were at 48 for ages, weren't we? And then, the little things sometimes. Anyway, so yeah, as Trudi says, the recommendations have been spread throughout sections 3 to 6. A lot more are found in section 4, genetic testing, and in section 6 for reporting, for obvious reasons. You will find in the publication that all the recommendations are in those grey boxes. That really helps them kind of stand out. And then we've put that must for an essential recommendation that we really think all labs that are doing HBOC testing should be doing. Must be doing, sorry, and then should, where it's a highly advised recommendation, but recognising that there are some circumstances and some labs that they may not be able to fulfil that recommendation. And those are highlighted in bold, but I've just put them in yellow for the purposes of this slide. So, pulling out some recommendations that we thought would be useful and particularly related to EQA and kind of some of the EQA schemes. I think, you know, we've been involved in marking EQA, so it's quite a good way of kind of seeing what some of the recurrent problems are. And so some of the key themes that are common to multiple sections is recommending referral to clinical genetics and having genetic counselling as an integral part of that genetic test. And the kind of the obvious scenarios, we've recommended that as predictive testing, where there's been a secondary or an incidental finding, whether that's via array or whole genome sequencing. And then if you're reporting a pathogenic variant, I'm using PV, pathogenic variant, but I also mean when it says PV, likely pathogenic as well, or a reportable VUS. Basically, if you think you're reporting something germline you think is associated, please refer to clinical genetics. Quality assurance is obviously a very key part of our jobs in the labs, and this is then shown again in sections 4 and 6, and it's things like making sure you've validated your tests appropriately, making sure you participate in EQA, like the EMQN schemes. So predictive testing, an example of this is making sure that you are testing the right gene region. So some of you will know, potentially being caught out by a case a couple of years ago where the assessors gave a legacy numbering for one of the cases. And some people worked out that that didn't really fit with the DNA sequence. Others just blindly kind of sequenced that area and didn't find the variant that was there. For section 6, making sure that you're reporting all that technical information. Us as assessors, we have to mark that when we're kind of scoring your EQA results, and so it's really important it's there, and that you're reporting the variant appropriately. Keeping up with new developments and guidelines sounds really kind of basic and obvious, but it was so apparent to us as authors that it's such a fast-moving field, and that just we were writing the guidelines and then there'd be a new paper out and I have to go back and edit and rewrite things. So it's, you know, it's amazing. There's lots and lots of work going on, and in particular these 3 areas. So the licensing and availability of PARP inhibitors, particularly for new cancers and particularly in different countries and jurisdictions, is massively in flux and changing, and you know, more treatment available to more patients, which is great news. The HBOC, the multi-gene panels, in the course of us doing the guidelines, we added RAD51C and D from the S2 list onto the S1 list for breast cancer, and variant interpretation. Need I say more? It's huge, and there's so much work going on in that, and it's definitely evolving and lots more to come. Okay, so now looking at section 4, genetic testing. So some of the key recommendations are around those multi-gene panels. So as Trudi said, we've put those kind of key, definitive, actionable genes in table S1, but please remember these are evolving, so it will be out of date, you know, in due course, so keep checking those guidelines. We've also tried to draw attention to some edge cases. Again, we've picked up via EQA. So, for example, the intronic variant, it's a pathogenic variant at -11. And it was really shocking how many labs did not pick it up because their bioinformatics pipeline stopped at -10. So we've put it in the guidelines to kind of try and build awareness and educate people. Also pseudogenes, I haven't got time to talk about that, but if people want to come talk to me about pseudogenes, it's one of my pet things afterwards, yes, please do. And one of the really important things that we kind of set out to do when we kind of formed the authorship group is as we wanted to define what we considered was a minimum testing service for HBOC. And we have defined this with lots of kind of consultation, collaboration, as sequence variants in BRCA1, BRCA2 and PALB2, and copy number variants in BRCA1 and BRCA2. We do still see a lot of reports from EQA that don't do copy number analysis. Due to the frequency of the variants found, particularly in BRCA1, we really feel you should be doing this testing. If you are not, then it's really, really important that you make that very clear in the report and you refer that patient on for further testing elsewhere. So again, another area of big flux was the treatment-directed somatic tumour testing. And if you're doing that testing, you really need to know that neoplastic cell content. And when you're thinking about doing the germline testing as well, we felt ideally you would do a paired germline somatic analysis. Obviously, that costs quite a lot of money. So I know a lot of people have opted to go for a somatic first approach. And if you do that, that at a minimum, if you find a somatic pathogenic variant with a high VAF, you should be confirming that in the germline because there is a high chance that that is germline in origin. And we know that the somatic samples and the pipelines are not good for picking up those larger indels and CMVs. So again, you might want to refer for germline testing in those cases. For targeted variant testing, it's again like the example I gave before, important you have a familial positive control, that you're SNP checking the primers and probes, and ideally you're minimising that area that you're looking at to reduce the risk of incidental findings. Again, we see kind of in EQA schemes in general, there is a question about a predictive test and someone does a whole NGS panel, and potentially you could find things that that patient has not been counselled for if you're doing that in a kind of real diagnostic setting. Okay, moving on to section 5, variant interpretation. So we're recommending that you use the ACMG guidelines, and included in that is the ACGS add-ons, and then for the gene-specific guidelines, the ClinGen VSIP, the Enigma BRCA1, BRCA2 guidelines, CanVig guidelines, and then the CMV guidelines. It's a bit of a nightmare how many guidelines there are at the moment, but obviously with the new ACMG version 4 coming out, it's hopefully going to simplify things, but it's going to take some time. We also refer to the non-coding variant guidelines, and we recommend that you submit your variants to database, particularly ClinVar. It's really important for that dissemination of information. We also recommend regular variant review, and in the paper we've set out the conditions when you would need to do that review, and we also give some guidance on reclassification of variants, which unfortunately has been happening a fair bit from this paper by Long et al. For somatic tumour variant classification, we recommend that you use the germline guidelines in addition to the somatic guidelines because so many are germline in origin. And then some things that are specific for HBOC. There's such a wealth of RNA-seq data, particularly for BRCA1 and BRCA2, and we know that there are these naturally occurring splice transcripts that can rescue your protein function. So even protein truncating variants in certain exons in BRCA1 and BRCA2, you can get alternative splicing, upregulated, and those are not pathogenic. So please consider this. Reduced penetrance variants. The example I've given here is the poster child of kind of BRCA1 reduced penetrance. We have a wealth of data on over 150 families that we know really clearly what the penetrance of this variant is. But Mother Nature is not like that. It's not black or white. So with missense variants in particular, we know that they have reduced penetrance compared to protein truncation variants, but there's some that are going to be up at the kind of truncating end, and there's going to be others that are much lower. So it's just worth considering and looking out for that, particularly if it's been identified in a Fanconi's case. For TP53 variants, you really need to consider CHIP, which is clonal hematopoiesis of indeterminate potential. I said that right, just about. And a big caution about reporting any missense variants in these genes that are primarily associated with truncating variants. And real missense variants are very rare in these genes. Okay, so on to reporting. So some of the essential must recommendations are you must state the variant guidelines used and your variant classification evidence. Also really important to state what variants you don't report, particularly if you don't report certain VUSs. Please include enough information about your technical details, including the test scope and limitations. So this is where you would put in if you do not do that CMV testing. Make sure that you're reporting the variant to the appropriate guidelines, and of course you're referring for genetic counselling. So some specific scenarios that also are musts is for TP53 and CHIP, you must state whether you've excluded CHIP, but also don't forget to look for the presence, whether it could be a real germline mosaic rather than CHIP. For cascades testing, predictive testing, make sure that you're looking at you state the region that you're looking at and any test limitations if you don't have a familial control. Somatic tumour testing, this does overlap with section 4, so I think I've kind of already covered those points. So we spent quite a lot of time on the reporting section and have tried to provide you a lot of information about reporting. I don't know if you can see the mouse. But okay, but on the left-hand side, it kind of shows you all the types of patients you might be referred to for genetic testing. So a diagnostic case, a case that, so potentially the first one with a family history, second line where somatic and germline PARP, where potentially no family history, but it's treatment directed, and then your familial testing. And then in the middle column, it's all the different possible outcomes you could get from that genetic testing. And then on the right-hand side, all the main points that you should cover in those reports. We've also included a little bit of guidance and some wording on some rare scenarios. So that's trans-heterozygosity in both BRCA1 and BRCA2, or rare biallelic cases with Fanconi's. And also in that, I'm sure everyone in the labs has come across it, where you have a familial variant, pathogenic variant, and you get somebody else from the family with breast cancer, and then they don't have that familial variant. And so we've again added some guidance for what you do in that scenario. And so we've also not just given the scenarios, we've also provided some example wording. Again, this is in the grey boxes and in italics, so you can choose to use that wording or bits of it, whatever you want. We've even provided the wording for a moderate risk gene, a reduced penetrance variant shown in yellow highlight. And the same for the predictive testing, and again, we've kind of separated out the males and given more information for reporting the males and depending on the gene. And I'm going to go back to Trudi, and I think we should get Stacey up here as well. Maybe.
Just to finish off with some final points. As Miranda said, towards the end, when we were trying to publish it, we really felt like this little lady here, trying to get to the finish line with all this new information coming at us constantly because it's evolving so much. So we did manage to capture a bit of a stop-start to, look at a new publication, decide, if it was relevant and incorporate it in if we thought it was. But then, obviously, since the publication, there are new guidelines coming out, and we know there are more important guidelines that are due soon as well. So this raised the possibility. We're exploring the idea of potentially, I think, Katie, it's your term, I think, a paperversary, where maybe after a period of time, we might just assess what new information is out there so that these guidelines don't drift into becoming completely out of date, that we can maybe, so there's something we're exploring at the moment. As we were preparing, actually, I noticed when we were preparing this presentation an error, so it's like an oh no, which we're currently in the process of correcting. So it's an error in one of the references, which is the same author, the same journal, but the wrong reference selected. So we're in the process of fixing that. So thank you, and if there are any questions.
So we're now going to look at the cardiac disorder testing guidelines, and Kate Thomson's going to come and give us a talk about this. So Kate Thomson did a DPhil with Professor Hugh Watkins at the Cardiovascular Medicine unit at the University of Oxford in 2018, when her research was focusing mainly on the genetic basis of inherited cardiomyopathies. She's continued her affiliation with the Radcliffe Department of Medicine at the University of Oxford and is involved in the Cure Heart Project and the Rare Disease Research UK Cardiovascular Initiative. She's again an HCPC registered clinical scientist. She's a fellow of the Royal College of the Pathologists, and she's got more than 20 years specialisation in inherited cardiac disorders at the Oxford Medical Genetics Lab. is where she's working currently. She's a member of the ClinGen Cardiomyopathies Variant Curation Expert Panel Leadership Group. That's quite a mouthful as well. She's a council member of the Association of Inherited Cardiac Conditions, and as well as her contributions to ClinGen, she's also contributed to the expert panel specification for the ACMG AMP interpretation guidelines for the sarcomere genes. So I'd like to welcome Kate.
Good afternoon, everyone. Thank you for the introduction, Nicola. So, my task today is to give you an overview of the EMQN recommendations for inherited cardiomyopathies and arrhythmias, which were published last year. So, for the rest of the talk, I'll refer to these as ICAs, hopefully. So this is our writing group. As the breast cancer group said, it's very much a team effort. Some of the members of the writing group are in the audience today. So we had 9 representatives from 5 centres across Europe and Australia, and they were there to share their experience and expertise in genetic testing for inherited cardiac conditions. We also had the wonderful Katie Shields from EMQN, who helped us steer the ship and kept us on track for quite a long period of time. I think these guidelines were over 4 years in the making. The draft document was also sent out to, I think, at least 85 laboratories who participate in EMQN schemes for community consultation, and we took into consideration their feedback in the final version. So what were the aims of the guidelines? Well, they were very much aimed at clinical laboratories providing testing for ICAs. And our intention was not to duplicate guidelines that were already there for clinical guidelines, variant interpretation guidelines, and reporting guidelines. What we were trying to do is pull together key information that was relevant to ICAs and help laboratories navigate the challenges of genetic testing in ICAs, hopefully facilitate best practice and improve the consistency of genetic testing for this group of disorders. So I'll walk you through the format of the guidelines, and I'll highlight just some of the key content and recommendations. So actually the initial remit of the writing group was to write best practice guidelines for hypertrophic cardiomyopathy and long QT syndrome. But when we first met as a writing group, we found that there was actually general guidance that were applicable to all ICAs. So to avoid writing 2 very similar documents with a few disease-specific aspects, we decided to write a general document with recommendations that were applicable to all ICAs, and then create disease-specific appendices for the most common conditions. So in the main document, we have general information relating to laboratory quality control practices, referral pathways, methods of analysis, results, interpretation and reporting, and also waving a flag for data sharing. And this is supplemented by disease-specific appendices for HCM, DCM, ARVC, long QT syndrome, Brugada syndrome, and CPVT. And the idea was that they would be kind of handy pull-out guides that would provide key information for each disorder. So I'll just explain a little bit more about the format and the content of these disease-specific appendices. So each appendix has a clinical summary section that gives a statement about the key clinical features of the disorder. We're also providing information on the age of onset and disease penetrance, prevalence estimates, and any relevant clinical or genetic testing guidelines for that specific disorder. We've got a section that's a genetics section that has a general summary about the genetic basis of each condition. There's also a statement about genotype-phenotype relationships that might be relevant to the laboratories when selecting a panel for testing or for results interpretation. And we also refer to any disease or gene-specific variant interpretation guidelines. There's a section on differential diagnosis, and this is really just providing information about other disorders that might present with similar clinical features and which genetic testing may be appropriate for. There's a genes table. Now, this is we wanted to include in this table the core set of genes which should be considered for testing for a specific disease area, and I'll cover more on how you choose those core genes later. But also in this table is information about the transcripts recommended for defining the regions of interest for variant calling and annotation. Where possible, we've aligned to the main transcripts, but for some genes we also included other clinically relevant transcripts. We've got a column about the predominant type of pathogenic variant in these genes, so whether that's truncating or non-truncating, information about the mode of inheritance for each gene, information about whether de novo variants might be found more frequently, so for example in RYR2-related CPVT. There's also information on the approximate diagnostic yield for each gene. In the table legend, we've tried to include other key pieces of information, so for example, regions of interest beyond the core coding regions, notable variants in those regions. So for example, in MYBPC3, one of the HCM genes, there's a variant that's at position -52, and it accounts for a relatively large number of cases, so it's important for labs to cover those regions. Also information about the presence of pseudogenes, so for example in filamin C, highlighting to labs that there's regions of that gene that you must be careful about when you're reporting variants because there's high homology with a pseudogene. And also isoform information, so for example for troponin T. For some of the appendices, there's additional tables that have information on the differential diagnosis genes that might be considered for inclusion in test panels. So for example, for HCM, might want to include the GLA gene, which causes Fabry disease and which can present with a phenotype that's very similar to sarcomeric HCM. And for some disorders, there's a table providing additional genotype-phenotype information that might be relevant for panel selection or interpretation or reporting. So on to the main document. So there are 7 sections in the main document. We've got limited time today, so I'm only going to focus on 4 points from the referral, analysis, and reporting sections. And I've chosen these because these were areas that we considered were important to cover in the document, but also some of these areas we found quite challenging to find a consensus on, so I thought it'd be quite useful to discuss it here today. So the referral section covers a few points, including the importance of a multidisciplinary team approach and the importance of undertaking testing in a laboratory that's specialist knowledge in ICAs, and the importance of pre-test counselling. But the area I particularly wanted to highlight today is the importance of having the right information at referral. So this is, in addition to having the, basic patient demographic information like age and sex, minimally, laboratories need to know what the headline diagnosis is, so hypertrophic cardiomyopathy or long QT syndrome as an example. You'd be surprised how often this information is not provided to laboratories. And obviously, we need to know that information to be able to activate the correct test. It's also really useful to provide information on age of diagnosis, which might not be the time the sample's actually sent to the laboratory. And that, again, for example, in hypertrophic cardiomyopathy, it might mean that a different panel is more appropriate, a panel with more early pediatric onset cases, for example. Also, additional clinical information that might be relevant, particularly if it's an atypical or unusual case, an earlier or more severe onset presentation. And there might be some specific phenotype information that's relevant to labs doing testing. For example, the presence of conduction disease in a patient with DCM, or skeletal muscle phenotype, or even deafness. Family history, again, really useful to have a pedigree, even if it's just showing first-degree relatives, and what is known, if anything, about their clinical status. And I think having this information really does allow laboratories to activate the most appropriate test in the first place, have the information that they need to interpret the results, and the information they need to write an accurate and clinically relevant report, and also recommend appropriate follow-up. So in the analysis section, again, there was a few points covered, but for today's talk, I'm just going to focus on the first 2 points. So which genes to select for the test panel and which panel to select for a patient. So which genes to test? And I think this point raised more discussion than any other area when we were creating these guidelines. And creating a definitive list of core genes that are recommended for testing is not as straightforward as you might think. But we were very lucky because when we were drafting these guidelines, several of the ClinGen ICA expert panel groups had been reporting their outcomes of their gene disease curations. So after much discussion, we decided that we would align our core gene list to genes that the ClinGen expert panel had assigned either a definitive or a strong classification, or had suggested that clinical testing would be appropriate. Although I do have to confess that there was a couple of bonus genes that managed to sneak their way in. A couple of these had not been part of the ClinGen curation, but there was quite strong feeling about that they should be included in these lists, and an example would be DMD in DCM patients. So in the main document, what we did do was stress that this list of core genes, that was the genes that was in the table 1 in the appendix, was not an exhaustive list, and that testing of other genes may be appropriate. But we reiterated that testing should focus on genes that had robust evidence supporting a gene-disease relationship. And if there wasn't an official curation by ClinGen or any other group, then we would recommend the use of published gene-disease validity frameworks, such as the ClinGen framework, so that labs can actually systematically and robustly curate the evidence prior to including genes on test panels. And I think just also making labs aware that there's ongoing gene curations. I think there's Jodie Ingalls has been leading one of the more recent HCM ClinGen gene curations, and hopefully those new genes will be published soon. Okay, selection of test panel. This was another area where there was a few differing opinions. So with current high throughput technology, obviously we can easily test more genes, and there's certainly demand and enthusiasm for large, broad gene panels, but there is a balance to be had. The more genes we test, and particularly the more genes we test that are not related to a patient's phenotype, we're going to find more variants of uncertain significance. And obviously in a clinical setting, these are problematic because there's a risk of misinterpretation and misdiagnosis. They create uncertainty for patients and the clinical teams managing their care, and they're very costly to follow up. So because these guidelines are for clinical laboratories, the recommendations we made was that to minimize the risk of identifying variants of uncertain significance, for straightforward cases with typical phenotype, select a disease-specific panel. And we would caution against the routine use of pan-phenotype, pan-ICA panels. I mean, obviously there will be cases where, atypical cases, or cases where there's multiple phenotypes in a family, and then of course it would make sense to apply more than one disease panel. Okay, the reporting, again, a few topics covered in this section, but the main point I was going to discuss today was the recommendations for familial and cascade testing. Sorry, I'm struggling to press the right button here. So one key component of the clinical report is advice around family testing, but there is often different advice given. So in these guidelines, what we wanted to do was recommend an approach that was based on variant classifications. So if a pathogenic variant has been identified in a proband, then molecular testing can be offered to the extended family, and the results of the analysis can be used to guide clinical management. But we did suggest a little bit more caution being applied if you're dealing with a variant that is considered likely pathogenic, where there's a bit less evidence. And in these circumstances, we would suggest testing clinically affected family members in the first instance before considering predictive testing. And this might provide more evidence, more segregation evidence, and it might change the classification of the variant. For variants of uncertain significance, we would not recommend testing unaffected family members, but it may be appropriate to test other clinically affected family members, again, to demonstrate segregation. With unaffected family members, it still may be appropriate to test parents if it's a gene where there's a high level of de novo variants, or there's any suggestion that there might be a variant might be de novo, so that would be appropriate. Okay, take-home points. So just going over today, what I've covered is the format and the content of the disease-specific appendices, the importance of having the right information at referral, the importance of selecting the correct genes that have robust disease gene association for the test panels, the importance of selecting a test panel based on patient phenotype, and then recommendations about family testing based on variant classification. There's obviously a lot more content in these guidelines, and please do go and have a look at the guidelines, and please do feedback any comments and suggestions you have, because again, like the breast cancer guidelines, these are going to evolve. We've already had the same thing with more publications coming out since we've published these guidelines that could be relevant going forward. And thank you for listening, and happy to take any questions.
Hi. Would there be any recommendation for laboratory to come back to clinician years after, once a VUS is not a VUS anymore?
Well, it depends which direction the VUS is. So I think there is definitely different guidance in different countries about how to proceed with them. The way we deal with it in our laboratory is if a variant is changing and it's a clinically relevant classification, we would contact the laboratory, and we are reissuing reports under those circumstances.
And not when it is classified as benign? Only when it is classified as clinically relevant?
So we would, if a variant, so the way we do it, and I guess other centres will be different, is if we have a VUS that becomes likely benign, we wouldn't necessarily contact the referring clinician then. And what normally happens is the referring clinician would contact us if they're about to see a family member, and then we would do a review and issue a report then. But we don't have the capacity to actively seek those. In an ideal world, I think these would be automatically updated and the clinician informed. But we would, if a variant changed from VUS to likely pathogenic or pathogenic, we would send out a report.
Right, so all it comes down to me to do is to give an absolutely massive thank you to the authors of the best practice guidelines. I mean, I was a clinical scientist from 1987 through to 2015 when I moved to EMQN, and I do remember how useful it was to have best practice guidelines to help you write your reports. And since I've moved to EMQN, of course, we also use best practice guidelines to help us mark your reports. So there's a very intimate relationship between EQA and best practice guidelines. So I'd really just like to say another thank you both to our authors who are here today, the authors who haven't come today, but also participated in writing the guidelines, and probably to a lot of you out there who've also contributed in our community feedback, because we think that's a really important part about writing best practice guidelines, is they have to be accepted by the community. So that's a really important part of the guideline process for us, is to do a community consultation, and then finally to have it published in a peer-reviewed journal as well. So thank you all for coming. Thank you all for your questions, and really a big thank you to Miranda and Stacey and Trudi, and I've just had a bit of brain freeze. And Kate. Sorry, I really am getting too old for this. So thank you all again. There's still some food left, and we've got a few minutes in the room, so please help yourself to any food. Have a chat with the guideline authors or any of us here at EMQN.